Hello everyone, and welcome to today's webinar, Generating Protein Reagents to Evaluate the Antibody and CD8 Positive T-cell Responses to SARS CoV-2. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar, as a part of the Cell Culture Heroes series, is presented by LabRoots and brought to you by ExpiCHO by Thermo Fisher Scientific. To learn more about the Cell Culture Heroes webinar series or stay connected on the latest events sponsored by ExpiCHO, please visit the Resources tab. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Natalia Guadalupe Herrera from the Albert Einstein College of Medicine. Natalia, you may now begin your presentation.
Hello everyone, and thank you for joining my webinar today. As was mentioned, my name is Natalia Guadalupe Herrera, and I'm currently a graduate student in the lab of Dr. Stephen Almo at Albert Einstein College of Medicine. And today I have the privilege to be talking to you about a project which really flourished during the pandemic, and it's titled Generating Protein Reagents to Evaluate the Antibody and CD8 Positive T-cell Responses to SARS CoV-2. And so a little bit about me before we begin. On the left is a picture of me, and on the right there's some contact information in case anyone has any further inquiries. My parents were born in Mexico, Michoacan. I was born in the United States. But growing up, I really didn't know a lot about research at all. It's actually one of the things that I really liked learning. So even though my parents didn't get a higher education, I worked really hard and got into college. And I ended up going to UC Santa Cruz and became an official banana slug. And yes, the mascot is a yellow banana slug. And along the way, I actually came across the STEM diversity programs at UC Santa Cruz. And these programs actually helped me get a foot into research and actually learn what research was and actually do the research, which is really interesting. So Mark, IMSD, and the UC Santa Cruz Genomics Institute really helped me along the way. I was also a part of the Howard Hughes Medical Institute for Exceptional Research Opportunities and the capstone programs, which actually added another layer and helped me do more research in another institution. So if there's any undergraduates here today or high school students and they're interested in research, I would encourage looking into programs that can help you get access to research and of course contacting the principal investigators themselves. So I did a lot of my undergraduate research at UC Santa Cruz with Dr. Victoria Auerbuch Stone. She studies the bacterial type 3 secretion system, which is here on the right. And it's a bacterial appendage that almost looks like a needle. And the way it works is pretty fascinating. It's able to inject effector proteins into host cells and actually manipulate host defenses in that way. And the interesting thing is that there's over 24 gram-negative pathogens that actually utilize this type 3 secretion system to cause disease, one of which is probably well known, which is the causative agent of the Black Plague. In this lab, we utilize Yersinia pseudotuberculosis as a model for studying the function of the type 3 secretion system, and this is a less pathogenic version of the plague. And I worked on screening the type 3 secretion system for bacterial virulence factors and characterizing a lot of the hits along the way. So along the way, I did get to meet a lot of very interesting people who helped me learn science, a lot of graduate students that helped me learn science and techniques and how to think about science. And I'm very appreciative of them here. So here's some pictures of our shenanigans. So I also was a part of the Howard Hughes Medical Institute XROP program, where I got paired up with Dr. Dan Lippman at the NYU School of Medicine. And along with my other mentor, Dr. Jason Hall, they really taught me how to think about science in a different aspect. So I was focused on microbiology, and they taught me a lot of immunology. And that's sort of what stuck with me, is that immunology was very interesting and something I wanted to pursue in my graduate career. And so now I'm at graduate school in the Almo lab at Albert Einstein. And I'm actually having the privilege of also getting to interact with and work with the Macromolecular Therapeutic Development Facility. Now, this facility shares the same lab space as the Almo lab. So the Macromolecular Therapeutic Development Facility, or MTDF, has a lot of nice resources that we can use, and I'll go through those resources in a second. My PI, Stephen Almo, is the scientific director. And one of the things we have access to as well, not just the physical resources, but a lot of the scientific intellect is there as well. So Dr. Scott Garforth, the operations director of the MTDF, has helped progress a lot of the projects in our lab as well. So the MTDF is pretty interesting because they have a lot of different services. They do high throughput cloning, expression profiling, so looking at bacterial mammalian proteins, large scale or high throughput production of proteins. And so it's of no surprise that the Almo lab is also having similar interests in designing and generating immunotherapy and immunotherapies that are protein-based, as well as high throughput X-ray crystallography and high throughput screening for finding novel binding partners in the human secretome. So I'll go through a little bit about the resources that the MTDF has to really look at protein production in a large scale. And so here's a picture of a robotics platform that allows us to liquid handle in a high throughput fashion. Here in the middle is a picture of a 96-well block where we're looking at, how we can grow up bacteria in a 96-well format. And then here on the right, we have a 2-liter bottle that's really looking at growth in a larger scale for protein production. We also have the capacity to purify proteins. And so here's a picture of one of the many FPLCs that we utilize in the lab. And just to show really the grandness of it all, like here's some gels. They're SDS page gels, they're protein gels. And we're seeing the proteins on several different SDS page gels. And these are just an idea of how many proteins we can purify in such a small amount of time. So using this prokaryotic platform, we've been able to purify thousands of proteins and solve hundreds of structures. Now moving on to the eukaryotic platforms. So not every protein should be expressed in a prokaryotic platform. This is obvious because some proteins require post-translational modifications, which are only present in the eukaryotic expression platforms and not in the prokaryotic platforms. So again, we have robotics, which allow us to set up, for example, hundreds of transfections in a few hours or hundreds of infections in eukaryotic cells. Here's an SDS page gel where we look at small-scale baculovirus expression of cytoplasmic adhesion proteins. And we can see that we can also do small-scale expression of secreted proteins in SF9 or Hi-Fi cells. And on the right, we're looking at small-scale expression in ExpiCHO cells, looking at an ICOS ligand-based immunotherapeutic. And so we really have different types of expression platforms we can use to look at different types of proteins. And of course, anything we can do in small scale, we can also do in a larger scale. So you see here another series of SDS page gels looking at the purification of several members of the immunoglobulin superfamily. course, we also can do large-scale expressions in Het293s with several proteins that are actually, some of them are immunotherapies, some of them are immunoglobulin family members that are Fc fusion proteins, effectively taking the Fc and dimerizing proteins together. And finally, on the right here, I just wanted to show an example of why we really like to use different expression platforms. So we have a gel where we're looking at the same immunotherapeutics, except on the left, we have expression of those immunotherapeutics in the XP293 platform. And on the right, we're looking at the same immunotherapeutics expressed and purified from an ExpiCHO expression platform. And what we can see is that on the right, we're really sacrificing purity of those immunotherapeutics because we're getting cleavage products. Whereas on the left, those same immunotherapeutics do not get cleaved in the XP293 platform. So really, this is one of the things that we can take a step back and see the same proteins being expressed in two different platforms, and trying to figure that out is one of the things that we like to do. So when the pandemic hit, the expertise of the MTDF and the Almo lab was really required because the scientific community needed to produce large amounts of spike protein from the SARS COV-2 virus. So I'll quickly just do a brief overview of the virus and why the spike protein is so important. So at the top, we have the SARS COV-2 virus, and we notice that there are some trimers, spike trimers, protruding from the SARS COV-2 viral membrane. The spike protein assembles naturally into a trimeric state, which is zoomed in at the bottom. And the spike monomers themselves can have two different conformations. So either the closed or the open conformation. And on the left, we're zooming into the closed conformation. What that means is that all the receptor binding domains are pointing down. So in the center, we have the receptor binding domains of each of the monomers of spike. And they're all being closed off, meaning the receptor binding domain is not accessible for binding to its receptor, which is ACE2 or angiotensin converting enzyme 2. On the right, we have sort of a conformational shift where one of the spike monomers, in this case, it's the cyan colored one, the receptor binding domain of that monomer is pointing upwards and now is accessible for binding to the angiotensin converting enzyme 2 receptor. And this interaction is very important in terms of SARS-CoV-2 infectivity because this allows entry of the virus into the host cell. So you can imagine the spike protein became an important target for biological, clinical, and structural investigations. So that's what part one of my talk will be focused on, is how we were able to generate large quantities of the spike protein and looking mostly at antibody responses of other biological applications as well. And so we'll start with the most obvious need for the SARS-CoV-2 spike protein, which is that we needed to test SARS-CoV-2 positive serum. So serological testing was important in the beginning for identification of potential donors of convalescent plasma. Additionally, we needed the spike protein ectodomain to identify therapeutic monoclonal antibodies. So how we did that was we identified single B-cell clones from convalescent patients. In addition, we also tried to use the convalescent plasma to then add into patients that were at risk of having a very severe response to SARS-CoV-2 to potentially, get them to not die. Biologically, the spike protein is very important because it allowed us to study the mechanisms by which B cells could respond to SARS-CoV-2 and what antibodies were being generated through the response. We also were interested in T cell responses, but that'll be more on the second part of my talk. And finally, we were also interested in structural studies. So we are a biochemistry lab that focuses on immunological targets. So one of our main interests was to do structural biology, and structural studies on the spike protein. And it became very obvious why it was so important. We needed to know how a spike protein binds to its receptor of interest, if we can actually find antibodies that could inhibit spike binding to its ACE2 receptor through antibodies targeting the receptor binding domain. And all this was really done by a lot of structural studies. And so that's what made it so important. And most of this work is already published, especially for part 1. So if there's anything that people would like to read in more detail, then please see our publication here. Now I'll get started with the outline. So first we're going to be talking about the protein production and functional validation. And we were looking at different spike ectodomain constructs. We decided to look at a few that were widely used, especially by scientists in our area for spike protein production. The first we termed OpSpike1, which was obtained from the McClellan lab. And there are a few key features of this spike construct, namely the spike ectodomain, which was cloned from amino acid 1 to amino acid 1208. The beginning part of the spike ectodomain is termed an S1 region, and the ending part of the ectodomain is termed S2. And I want to direct you to a few interesting parts, namely the receptor binding domain region, which is an S1. An additional interesting region is the S1-S2 cleavage site, which for the purposes of our studies and for the purposes of a lot of those structural studies, scientists didn't want them to be cleaved. So the RR8R region that was recognized as a polybasic furin region was actually mutated to a GSAS to avoid cleavage by S1-S2 proteases. Additionally, at the very end, after the spike ectodomain, there's a T4 fiber chain trimerization motif, and that was utilized to actually get the spike monomers to trimerize in solution. Additionally, this specific OpSpike1 has a protease site which gets rid of the 8X his and the twin strep tags. For OpSpike2, which we obtained from the Kramer lab, it is actually very similar to the OpSpike1 protein, except for a few differences, namely the length of the spike ectodomain goes from 1 to 1,213 amino acids. The mutation to mutagenize the polybasic furin site is actually truncated to an A, and the thrombin cleavage site actually gets rid of the T4 fiber chain trimerization motif, and there's a 6X his tag at the end. But otherwise, they're pretty similar. And so we decided to use both of these and purify the proteins using nickel affinity chromatography and size exclusion chromatography, which we will not be showing that type of data today. And because the spike protein is highly glycosylated, we didn't want to use prokaryotic systems. We wanted to use eukaryotic systems to allow for those post-translational modifications to be included. So we used two systems to produce the spike protein. On the left is the XP293 transfection kit, and on the right is the ExpiCHO transfection kit, which we used for both of these constructs. So what happened when we tried to purify and express the OpSpike1 and OpSpike2 using ExpiCHO and XP293 systems? Well, on the left is a gel, an STS page gel. Lane 1 and lane 2 is OpSpike1 protein. Lane 3 and lane 4 is OpSpike2 protein. Lane 1 is the OpSpike1 protein produced in ExpiCHO, and lane 2 is the OpSpike1 produced protein in XP293. And one of the things that we noticed is that there's a slight difference in migration on the STS page gel. And this was reproducibly true every single time we produced the proteins. And one of the hypotheses was that there's a difference in the glycosylation in XP293 versus ExpiCHO. So we actually did look at the types of glycosylation adducts that were present in ExpiCHO-93 and ExpiCHO-produced OpSpike1. And we found that there were notable differences in the types of adducts present, but we will not be discussing these data today due to time constraints. But we found that it was very important that the spike protein can be glycosylated and hyperglycosylated. And there are a few studies showing that it can actually possibly even affect antibody binding because the glycan adducts are very big and can mask binding sites of antibodies. So glycosylation of spike is a very important topic. We just do not have time to go over it today. What we were interested in is also the expression yields out of these two systems, comparing OpSpike1 versus OpSpike2. And what we saw was that overall, OpSpike1 expressed better compared to OpSpike2. So we see the first two bars are OpSpike1 produced in ExpiCHO-293 and OpSpike2 produced in ExpiCHO-293 in red. And it was much greater yield for OpSpike1 produced in ExpiCHO-293 versus OpSpike2. And the last two bars, the green and the purple bars, are OpSpike1 and 2 produced in ExpiCHO-S. What we can see is that the green bar, the OpSpike1 protein produced in ExpiCHO, produced the highest yield out of all the different variations, leading us to almost 30 milligrams per liter of protein. And this was very important to us because we knew we had to make a lot of protein, a lot of the spike protein. So we're trying to figure out what was our best cost-effective way to make a lot of protein using less liters of cell culture. So we ended up making 170 milligrams of OpSpike1 protein generated from these ExpiCHO cells. And this was compared to the reported yields of about 0.5 milligram per liter in HEC-293 freestyle cells at the time. So one of the things that I'd like to point out is that there's differences in expression systems, and they do have an effect on the protein glycosylation, stability, and yield. And finally, of course, you make a protein. If there's anything I want to leave you with is that you have to test the functionality of the protein you make. So we did this. We expressed human ACE2 with the C-terminal GFP tag in HEC-293F cells. We then added in our trimerized spike that has an 8X His tag, and we could detect binding of the spike protein to these cells using an anti-His secondary antibody that has a PE conjugation. And we did this by flow cytometry. So on the y-axis, we have the geometric mean of the PE signal from the antibody. And on the x-axis, we have increasing concentrations of the trimerized spike protein. And the black line represents binding to the human ACE2 expressing cells, whereas the pink is the mouse ACE2 expressing cells at the highest testing concentration, which shows that there's no binding as expected to mouse ACE2, but there is binding to human ACE2, indicating that we purified a functional, non-aggregate spike protein. And so then we proceeded to do some structural characterization. So we decided to do cryogenic electron microscopy and single particle reconstruction to assess the tertiary and quaternary assessments of the S protein. And we actually did this for both ExpiCHO-293 and ExpiCHO expressed OpSpike1. But here I'm just showing the OpSpike1 produced in ExpiCHO. And this was done with Dr. Jeffrey Bonanno from the Almo lab and the New York Structural Biology Center. And there was, at the time, a lot of reported SARS COV-2 spike Cryo-EM structures displaying the trimeric spike existing in both a closed conformation, so all the receptor binding domains pointing downward, or there was also a lot of heterogeneity where the spike monomers were also in the open conformation. So you had a lot of structures that had one spike monomer with the receptor binding domain pointing upwards. We're here just showing the one spike structure we solved, which is the symmetrical trimer. So all the spike monomers have the receptor binding domain pointing downward. And this was the easiest structure to solve. So we did this at a 3.22 angstrom resolution, and we inserted our structure as a PDB file 6x6p. On the left, we have the side view of the spike trimer, with each monomer colored in a different color, so black, gray, and then the purple ribbon. On the bottom is, you're looking at the viral membrane, and the very top part is where the S1 is, so where the receptor binding domain and the N-terminal domain are. Closer to the viral membrane, you have closer to the S2 region of the spike ectodomain. And if we look at the top view, so we're looking at the very top of the spike trimer, we see that, again, the whole trimer is symmetrical, and each monomer is colored in a different color. But we didn't just want to look at the spike trimer and do a cryo-EM structure on it. We also wanted to look at how this compares to all the solid structures at the time. So we did an analysis of all the cryo-EM structures solved at the time, and this was just looking at all the spike trimers that were symmetrical, meaning all the receptor binding domains were pointing downward. And so there was 20 cryo-EM structures that we aligned to PDB file 6VXX. And what we noticed is on the left, when we're looking at the side view, they all agreed pretty well with one another, which was pretty fascinating that 20 different labs could generate all of these cryo-EM structures, and they could all agree with each other. The S2 region seems to be less variable, while the S1 region, closer to the top, seems to be slightly more variable. And if we look at the top view, we really see the variability, the receptor binding domains in the center and the N-terminal domains on the outside of the top view, where you can see that there's generally more variability compared to the S2 region, but they seem to agree with one another overall. When we looked at all the 20 cryo-EM structures, we actually saw something pretty fascinating, which is if you look at all of these cryo-EM structures, you see that there's two distinct conformations in a particular region, and that's in the region of amino acids 614 to 642. So this is conformation 1 on the left, where we're really highlighting the region of interest 614 to 642 in magenta. And you'll notice that there's red stars. This indicates where there's missing amino acids, and that could be due hypothetically to variability or disorder in those regions, so they couldn't map the region. So they're missing amino acid 621 to 640 in this case. But looking at 9 structures, the region of interest in magenta aligns pretty well, and it aligns to the specific region of conformation 1. But the second set of structures that we looked at, and there were 7 of the cryo-EM structures that fit conformation 2, and you'll notice that the magenta region is now pointing in a different area, and it has a different conformation. And these set of structures were missing amino acids 619 to 631, except for one of the structures, which actually did model the whole entire region. Another thing that's very interesting, when we looked at conformation 2, we found that the region highlighted in blue, right under the magenta region, which constitutes amino acids 828 to 854, was not present in conformation 1, and in conformation 2, this region seems to be pretty ordered. So this is very exciting because it suggests that the conformation that these structures take really affects this other separate segment. So what we found is that there were 2 conformations, distinct conformations. Our model actually seemed to display conformation 1, which is very exciting. But Spike seems to exhibit at least 2 different conformational states for the segment containing amino acids 614 to 642. And this seems to affect the stability or the ordering of amino acids 828 to 854. And this was pretty interesting to us, and the biological implications of these conformational states has yet to be studied. And so lastly, I want to go through biological applications. So there are a few that I'm very interested in. Our lab has developed a multi-antigen protein array, which I'll discuss first. Dr. Alev in our lab has developed this SARS COV-2 multi-antigen protein array, where we take SARS COV-2 purified antigens and we cross-link them to aminosalicylic glass slides. We then add in human serum to the slides, and if the human serum contains an antibody that can bind to the antigen, for example, here in this picture we have spike protein, then we can detect that bound antibody using an anti-human IgG antibody. And we can do this not just for IgG antibodies, but for IgA and IgM isotype antibodies. So the SARS COV-2 multi-antigen protein array looks something like this. On the right, we have a SARS COV-2 negative serum, where we really only see two signals, the human IgG control at the very top, and the human IgA control at the very bottom, boxed in green. We also have two SARS COV-2 serums here, just as an example, where we see that we get much more signal, where we see full-length spike boxed in yellow, S2 region, S1 region boxed in blue, the receptor binding domain, nucleocapsid, which is boxed in white at the very bottom, and the mutant version of spike, which is D614G, which is boxed above nucleocapsid, that also seems to have very bright signal for these two serums. So we have a lot of options with this type of protein array and this type of platform to screen for different antigens, different spike variants, and look at different types of antibody responses. And I'm just going to show a quick overview of what this would look like. So here we're just looking at two positive serums, serum 12 and serum 15, which are SARS-CoV-2 positive, and a SARS-CoV-2 negative serum, which is in green. And we're looking at IgG spike antibodies, spike IgA antibodies, and spike IgM antibodies. And what you see is that overall, serum 15 has a higher antibody response compared to serum 12. And on the x-axis is where we're looking at the log dilution of the serum. So the more antibody you have, obviously the more response signal you'll get. On the bottom, we're looking at the receptor binding domain binding. So receptor binding domain IgG antibodies, IgA and IgM, and of course, serum 15 has a higher antibody response for everything than serum 12. And what she's really trying to do is see how these levels of antibody responses correlate with overall clinical outcome for these patients. So as you can tell, this platform is very exciting because it allows us to characterize the antibody responses to SARS-CoV-2, but also we have a platform that allows us to characterize antibody responses to any type of viral infection. So next I wanted to talk about another platform that's in our lab that's very exciting, which is a high throughput screen of the Human Secretome. And we wanted to look at novel spike binding partners. So what we did is we took a library expressing several types of C-terminal GFP fusions, but here in my cartoon, I'm just showing ACE2, human ACE2 as a positive control, where human ACE2 expressing cells will bind to the trimerized spike at 500 nanomolar concentration, and we can detect this binding using an anti-His secondary antibody that's PE conjugated, and we can detect this by flow cytometry. So looking at our GFP expression library, we were able to do high throughput, small scale hectogen 3 freestyle transfections of our library members, wait 48 hours, and then add in recombinant spike 1 to the transfected cells. We then added in our PE conjugated anti-His, and we performed high throughput flow cytometry to determine whether or not any cells would bind to our trimerized spike. And as you can imagine, we did this with spike protein, purified spike protein, but we could have also done this with cells expressing spike or any other target of interest. So when we did this, we were first looking at a few main targets of interest. human ACE2, which is our positive control with the geometric mean on the y-axis. Mouse ACE2, which does not have signal, indicating there's no binding, as our negative control. And there were two receptors present here that were of interest to us at the time. CD147, which was actually a proposed binding partner for spike at one point, and has now been shown it is not by several lab members in several different labs, including ourselves. So we did indeed not find binding of CD147 to spike. We also didn't find binding of spike to CD26, which is a receptor for MERS coronavirus, and we did not find binding for that as well. Looking at our high throughput screen results, we screened 20% of the human secretome to look for novel spike binding partners, and we found nothing. So in green, we have binding of spike to human ACE2 expressing cells, which was always our positive control to ensure that our spike protein was working properly. And in purple, we have Fc receptor expressing cells, which are able to bind to the antibody we add to detect binding. So these were also false positives. A library consists of immunoglobulin superfamily members, TNF receptor superfamily members, integrin family members, GPCRs, and chemokine receptors, just to mention a few. And we did not detect binding of spike to any of the receptors that at the time the literature was saying spike could bind to. So integrins, CD147, Siglec-9 and Siglec-10, CCAM1 and CCAM5 were all at one point speculated or thought to have bind to spike, and we showed that that was not the case. But this platform is very interesting because it allows us to find binding partners in a high throughput fashion, not just for SARS COV-2 spike, but for any type of other ligand or protein of interest. And so I'll quickly summarize part one. We have an optimized spike protein production protocol where we actually express large amounts of spike protein in ExpiCHO cells. And we showed the spike protein using our purification scheme is of good quality and is functional. And spike protein produced in ExpiCHO cells is differentially glycosylated compared to the published analysis of spike protein produced in HEC293 freestyle cells, which we just didn't have time to show today, unfortunately. But we also see that spike protein can be utilized for several downstream applications. So we screen, and we're currently screening for antibody responses to patients that have been exposed to SARS CoV-2. We're looking at IgG, IgM, and IgA antibody responses to several SARS CoV-2 antigens, including spooling spike. Screening for spike interactions with all the receptors in the human secretome has thus far yielded no new interactions, but I'm still a big fan of this platform, so I wanted to talk about it. And the spike protein has been utilized by a lot of our collaborators for clinically relevant experiments looking at convalescent plasma, B-cell sorting, and so I just did not have time to go over all of this data today, but spike protein does remain relevant for these types of analyses. The second part of my talk, which is a smaller portion, is looking at whether or not we can modulate SARS CoV-2 CD8 positive T cell responses. And so when you have a SARS CoV-2 infection, there are different arms of the immune system that play an essential role. You have the innate immune system, but you also have the adaptive immune system, which also plays a significant role. And a lot of scientists have focused on the B cell arm, where you have plasma cells that secrete antibodies that can neutralize SARS CoV-2 and prevent entry of SARS CoV-2 into host cells. And while that is very important, we in our lab are very interested in looking at the T cells and specifically the cytotoxic T lymphocytes. And these are important for finding virally infected cells and killing them. So any cell that would be infected with SARS CoV-2 would have to interact with the cytotoxic T lymphocyte, and the cytotoxic T lymphocyte should recognize and kill these virally infected cells. So one of the interesting things that we found, there are a few publications out there suggesting that unexposed individuals have CD4 positive and CD8 positive T cells that can actually respond to SARS CoV-2. And the hypothesis being that these people have been exposed to common cold coronaviruses that have a high degree of conservation in some parts of, spike or some parts of viral proteins. And these viral proteins were presented to CD4 and CD8 positive T cells, and these T cells can now respond to SARS CoV-2 because of that. And an additional hypothesis that several scientists have, including our lab, is that those individuals might have a milder response to SARS CoV-2 because of previous exposure to common cold coronaviruses. So our goal is whether or not we can actually activate those SARS CoV-2 CD8 positive T cells specifically, because CD8 positive T cells can find and kill virally infected cells. And we want to see if we can activate these CD8 positive T cells in unexposed as well as exposed individuals to prime the immune system before SARS CoV-2 exposure. So what would it look like to get activation of a CD8 positive T cell? We have a schematic on the right where we're looking at CD8 positive T cell on the right, on the top, expressing a T cell receptor. Now, on the bottom, we have an antigen presenting cell, which is going to do exactly what the name says, which is present antigen. So let's say that cell itself, the antigen presenting cell, took in virus, and especially SARS CoV-2 virus, and chopped up the proteins and now is presenting a SARS CoV-2 peptide onto its MHC class 1. If the T cell expresses a T cell receptor that can bind to this MHC peptide combination, then it can start to become activated. But it needs an additional co-stimulation or a secondary signal in order to become really activated. So here I'm just showing an example where we get IL-2 co-stimulation. But co-stimulation can occur in different ways. IL-2 can activate T cells. CD28 can activate T cells. So there's different ways that we can activate T cells. This is just one example. But what we like to do in our lab is we developed a novel class of immunomodulatory proteins, and they're termed SynTacs. They're artificial immunological synapses for T cell activation. And this is just a protein reagent that we're using to mimic the antigen presenting cell. So what we have is the MHC class 1 in pink with the peptide in orange. And we also have it linked to something that can modulate T cell activation. So IL-2 or something that can activate CD28, for example. But what happens here is that the CD8 positive T cell has a T cell receptor that can interact with the MHC and peptide. So this will activate the T cell receptors and giving the T cell signal 1 to be activated. You can also get a signal 2 or a co-stimulatory signal to further activate that T cell by giving it IL-2. And using these SynTac biomolecules, we can activate T cells in an antigen-specific fashion, because now we're only activating the T cells that have a very specific T cell receptor. And so what that T cell would do when it's activated is shown on the left. So that T cell that expresses that very specific T cell receptor is now able to detect when an infected cell has been infected with SARS CoV-2. Because the SARS CoV-2 infected cell will process SARS CoV-2 antigens, present the peptides onto MHC class 1, and if the CD8 positive T cell expresses a T cell receptor that is capable of binding this complex, then it will perform an immune attack and kill the infected cell. And that's exactly what we want. And really something that we want to do is not just activate one specific CD8 positive T cell with one specific T cell receptor clone. We want to make an array of different SARS COV-2 epitope syntax to target several different types of CD8 positive T cells that can all respond to SARS COV-2 peptides. So the goal is to really do this, to activate CD8 positive T cells and have them be activated towards SARS COV-2 infected cells. Now, do these SynTac biomolecules work? We have evidence to show that they do. So looking at an HIV infected donor, we were able to take out HIV PBMCs, or peripheral blood mononuclear cells. These peripheral blood mononuclear cells contain several types of cells, including CD8 positive T cells. But all of these cells get injected into a non-SCID gamma mouse. This non-SCID gamma mouse actually is very immunodeficient and lacks mature T cells, B cells, and natural killer cells. So this allows us to engraft primary human cells into this mouse model. We actually take the mouse spleen after 7 days and do flow cytometry analysis. And we do this for either untreated or mice that were treated with a SynTac biomolecule. And this SynTac biomolecule is targeting the HIV-specific CD8 positive T cells. Now, this is the data. So on the left, we have staining of an untreated mouse splenocytes, where we're looking at the CD8 marker on the bottom axis, on the x-axis, and the SL9 tetramer on the y-axis. So this SL9 tetramer is a protein reagent that can bind specifically to the HIV peptide, to the T cell receptor expressed on T cells, that can respond to the HIV peptide termed SL9. So we see that we have about 0.35% of the total cells are specific for this HIV peptide. So only 0.35% of the T cells of this HIV-infected individual can respond to HIV-infected cells. In the middle, what we have is actually the same type of staining experiment, but this was after treating with our HIV SynTac. So now the goal is, can we activate and proliferate these T cells in the mouse? And the answer was yes, comparing untreated to the SL9 anti-CD28, where we have the SL9 HIV peptide loaded on MHC, which will specifically bind to and activate HIV-specific T cell receptors. And we have anti-CD28, which can activate CD28 molecules on T cells to further stimulate T cell activation. What we see is we get 12.1% of the population is now that specific T cell that can respond to HIV-infected cells. We've expanded that population. And now we're looking at a negative control, which has the incorrect peptide. It has a CMV peptide loaded onto MHC class 1. And we see that the HIV-specific T cells do not have the T cell receptors to respond to this SynTac, so we should get no expansion. And indeed, if we look above at the staining, we do not see that that's the case. So if we look at the quantification, the HIV-specific anti-CD28 SynTac in the middle activates CD8 positive T cells in an antigen-specific fashion. So we got expansion of these T cells in an in vivo format. And actually, we've shown that these T cells are active towards HIV-infected cells. And for lack of time, we will not be going over that today. So it's very exciting. We've shown that SynTac biomolecules can activate viral-specific CD8 T cells. So we have premises to believe that we can use this platform for SARS COV-2 as well. And so the current work in the lab is to generate a variety of these SARS COV-2 SynTacs. So on the left is an SDS page shell of one peptide in the SynTac platform in a small scale initial test. And we saw that we get protein, which is very exciting. But of course, we want to take advantage of all of the resources we have through the MTDF to screen for greater than 52 different SARS COV-2 epitopes in the SynTac platform. And using different types of cell types, ExpiCHO and ExpiCHO-93, to optimize expression of these SynTacs. And with that, I'd like to end part 2, where we're really doing, this is really work in progress. We're looking at optimizing the expression and purification of these SynTac biomolecules in several different cell types, ExpiCHO, XP293, and HEC293. And we're really going to start looking at trying to expand these SARS CoV-2 specific CD8 positive T cells and see if we can activate them towards SARS CoV-2 infected cells in either exposed or unexposed individuals to SARS CoV-2. And with that, I'd like to thank all of our collaborators. There's a long list of collaborators here. I'd like to thank Albert Einstein College of Medicine, Montefiore, Q Biopharma, who's doing the SynTac work, and Thermo Fisher Scientific for this wonderful opportunity. And with that, I'd like to take any questions.
Thank you, Natalia, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how effective is the SynTac at activating antigen-specific T cells?
Okay. Hi. Yeah, excellent question. So the SynTac themselves are pretty effective at least the ones that we've tested at activating T cells. So here I'm just showing a slide where we're looking at markers of activation. So the T cells would be either secreting TNF alpha and interferon gamma. And to compare, in the immunological field, a lot of people use an anti-CD3, anti-CD28 antibody for general activation of T cells. So that's what we're comparing it to. And if we're looking at our antigen-specific activation, where we're only activating the T cells that can respond to the SL9 HIV peptide with an additional CD28 activation attached to the SynTac, we see that we can activate those T cells pretty well. And we have differences in activation mods, so like activation with secondary molecules, so CD28 or 4,1-BBL. So yeah, they're very effective. And if we look down below the TNF alpha, interferon gamma, we also see perforin and CD107A markers. So those are also pretty, those T cells are staining pretty well after SynTac treatment. So yeah, they're pretty nice and effective.
Thank you, Natalia. Our next question, can you use the high throughput secretome screen for the identification of novel interactions in disease contexts? For example, screen for binding partners to tumor cells.
Yes, so if we look at our slide that was shown previously, we were using purified spike protein, and but this isn't to say that we can't use other types of ways to present ligands or receptors of interest. So we have done this type of screen with cells expressing ligands, and then we screen against the library of cells. And much like that, you can also take a cancer cell line of interest and see what you can find for binding using this high throughput platform as well.
All right, thank you again, Natalia. Our next question, can you utilize the SARS CoV-2 multi-antigen protein array to screen for differences in antibody responses to SARS CoV-2 spike variants? For example, can we measure if there is a notable difference in antibody responses to the Delta variant?
Yes, so going back to this slide, we were able to use our SARS CoV-2 antigen multi-protein array to look at different spike variants as well. Some of the notable ones that we have used
is the N501Y mutation, the D614G mutation, and then the Delta variations as well as others. And that's work that's currently done in our lab, and we'll be trying to get it published soon. But yeah, that's a great question.
All right, well, thank you, Natalia. That's all we have time for today. Do you have any final comments for our audience?
Yeah. I guess if anyone's interested in our lab, reach out to us. Our lab is looking for technicians for the MTDF and for all postdocs who are interested in this kind of work, specifically the SynTac platform. And yeah, any other questions that were pressing, feel free to contact me since we didn't get to all the questions today.
Thank you again, Natalia, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, GIBCO by Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.